Hi everyone, in this video we'll be explaining about Calvin cycle. You guys might be wondering what really is Calvin cycle. For your information, Calvin cycle is about how plants fix carbon dioxide. In the previous topic, we have learned about how NADPH and ATP were produced. But in this topic, we're going to explain how carbon dioxide is converted into organic molecules or named as glucose. In short, the whole process can be seen in this chemical equation that we have shown in this video. Calvin cycle will be divided into 3 stages. Firstly, about carbon dioxide uptake. Secondly, carbon dioxide reduction. And lastly, REBP regeneration. Okay, let's go! The first stage of Calvin cycle, which is carbon dioxide fixation. What is carbon dioxide fixation? It is when CO2 reacts with 5-carbon ribulose biphosphate or RUBP. This reaction catalyzed by an enzyme called Rubisco. This reaction will form 6-carbon product which is unstable. So what will happen when it is unstable? It will break down or split into 2 molecules of PGA. So in summary, CO2 reacts with RUBP catalyzed by an enzyme called Rubisco and will form 2 molecules of PGA.
Although Calvin cycle is not directly dependent on light, it is indirectly dependent on light since the necessary energy which refer to ATP and NADPH are products from light-dependent reaction. ATP from light reaction provides energy to produce chain of phosphoglycerol. This is called reduction reaction because it involves the gain of electron by PGA. The electron and hydrogen comes from NADPH. ATP and NADPH are used to reduce 12 molecules of PGA into 12 molecules of glycerate triphosphate. 2 molecules of glycerate triphosphate leads to formation of glucose. ATP and NADPH that were used lost energy which resulted both of it to convert into ADP and NADP+ to be reused and re-energized.
Now we're going to talk about RUBP regeneration. Firstly, 10-GGP, which is 30 carbons, remains in the Calvin cycle and actually went into reactions to form 6 molecules of ribulose phosphate, or we can call it SRP, which contains only 1 phosphate, and actually went into a reactions called phosphorylated. Phosphorylated is when you take the phosphate from the ATP to form a 6 REBP, which is ribulose biphosphate because now it has 2 phosphate, and REBP is used to restart the cycle, and the REBP regeneration is done.
Now, let me recap again the whole process of Calvin cycle. The Calvin cycle begins with the carbon dioxide uptake phase. Here, 6 molecules of carbon dioxide are captured by 6 molecules of ribulose biphosphate, RUBP, a 5-carbon sugar. This reaction creates an unstable product that immediately breaks apart into 12 molecules of 3-phosphoglycerate, PGA, each containing 3 carbon atoms. Now, in the next step, known as the carbon reduction phase, each of the 12 PGA molecules is phosphorylated by ATP and reduced by NADPH, forming 12 molecules of glyceraldehyde, 3-phosphate, G3P. 2 molecules of G3P are then used to synthesize glucose and other carbohydrates. The remaining 10 molecules of G3P move to the next stage, which is the RUBP regeneration phase. During this phase, the 10 G3P molecules are rearranged through a series of reactions in order to regenerate 6 molecules of ribulose phosphate, which are then phosphorylated to form 6 molecules of RUBP. With the regeneration of RUBP, the Calvin cycle is ready to start again, capturing more carbon dioxide and continuing the process of carbohydrate synthesis.
How about the importance? There's a few importance of Calvin cycle. 3-GP or glyceraldehyde 3-phosphate can be converted to many other molecules. The hydrocarbon skeleton of 3-GP can form into banyak benda, iaitu fatty acid and glycerol to make plant oils, then glucose phosphate or easily known as simple sugar, then fructose. Fructose ni bila dia bergabung dengan glucose, dia akan menghasilkan sucrose. Next, starch and cellulose, and lastly, amino acids.
Thank you. Bye.